Welcome to this live from the field event. This is about chemistry in ecology, and we give students a window into the process of scientific inquiry and get to hear from researchers and professors spanning 3 countries. We'll find out who they are, how they got interested in this field, hear about their research, and learn about how students can engage. My name is Kerry Wininger with Sonoma State University's Center for Environmental Inquiry. You can learn more about us at cei.sonoma.edu. For instructors in the audience, be sure to take a look at the document they'll be pasting in the chat. It has extra readings and exercises, and also visit thevirtualfield.org to view similar past events. So here are some guidelines for the webinar. Since it's a webinar, all participants are muted. Your video cameras are off automatically. If you'd like closed captions, you should have the option to activate that in the toolbar as an icon or by clicking the More icon. Please use the chat button to communicate, and please submit questions for the presenters using the Q&A button instead of the chat. Also, please note, if you're a student, make sure your full name and the word student is visible somewhere, either as your username or typed before your question. That way you receive attendance credit and your question is prioritized. We are so fortunate today to have as our moderator someone with experience in both field research and education. Dr. Martha Groom is a professor in the School of Interdisciplinary Arts and Sciences at the University of Washington, Bothell. In addition to over 30 years of teaching undergraduate ecology and conservation, Martha also implemented 4 OCELOTS modules, including one about gold mining in the Amazon that will be presented today. So without further ado, I will turn this over to Martha.
Thank you. And thank you all for joining us. As you'll see today, in each of these live from the field events, we virtually visit with field researchers, educators, and students at 3 different locations. These events are a project of the Virtual Field. It's an international coalition of 50 field stations and marine laboratories supported by the Organization of Biological Field Stations and by the National Science Foundation. Other virtual learning experiences help college students build observation, communication, questioning, and data analysis skills through 360-degree videos, which are awesome, underwater film, and more. You can learn more at thevirtualfield.org, and Kerry's put that in the chat. This is the last of 4 special events that are taking place in collaboration with the OCELOTS network, online content for experiential learning of tropical systems, or OCELOTS. It brings together researchers, active learning pedagogy specialists, software developers, and media specialists to create an open access online resource library of learning modules in tropical biology. The goal is to spark excitement in undergraduate courses by broadening cultural and geographic perspectives, enhancing principle-based reasoning and quantitative skills, and changing the way students learn biology. You can learn more at the link that Kerry's put in the chat. Today, we'll take a look at some ways that chemistry plays a role in ecological studies, talking to both the folks who have been in the field collecting data and the educators bringing those data into the classroom through the OCELOTS modules. Speaking from my own experience, one of the best aspects of ecology education is getting to expose students to exploration of and data collection in different ecosystems. And that's a key aspect in fostering student interest in the field of ecology. Today's topic of chemistry and ecology is interdisciplinary in nature and can get us thinking about how what we are learning matters in biology and chemistry, conservation, and beyond. We hope to expand your vision in thinking about how working across different disciplines enriches our understandings, with a focus today on how gold mining in the Peruvian Amazon affects the bioaccumulation of mercury, and at the organismal level, how chemistry may influence food choices of seed-eating animals and hence plant survival in a Costa Rican rainforest. I became involved with OCELOTS personally because of my experience and love for tropical ecosystems. I began serving as a research assistant in the Peruvian Amazon right after college, which led to my completing my master's in the same location, Manu National Park. And then I took classes and conducted research in cloud forest and lowland rainforest of Costa Rica. Later, I supervised a group of student researchers in coffee agroecosystems in Puerto Rico, and I was very lucky to have the opportunity to work in tropical ecology in all these ways. However, I know many of my students won't be able to experience these ecosystems firsthand. So I was super excited to use the tropical forest ecology modules from OCELOTS in my ecology classes for folks who haven't traveled outside the US. These modules are really a great way to expose students to ecosystems and areas of biology that they wouldn't otherwise know about and increase their interest in these fields. These types of online learning modules humanize the scientists and celebrate collaboration and research all over the world. Today's event features 3 teams of biologists, 2 who created modules based on their research, and 1 who implemented a module in the classroom with her students. We'll paste a link in the chat now to access the modules covered today, along with implementation plans and associated readings. Each team has prepared an 8-minute video describing their work and experiences, and we'll play these for you. After the videos, they'll all be available to answer your questions live. I encourage everyone to post your questions in the Q&A as you think of them, right as you think of them. And again, post in the Q&A. Kerry and I are going to gather up your questions, and we will ask them out loud to the presenters during the panel discussion portion of the event. So for now, we will segue into our first video. So our journey starts in a territorial reserve in Madre de Dios, southeastern Peru. Dr. Jacqueline Gerson from Duke University and her colleagues conducted research on the impacts of gold mining on species in the Amazon. Dr. Ursula Valdez from the University of Washington, Bothell, and Dr. Roxana Arauco of the Cochacashu Biological Station have worked extensively in these same sites. And they created the OCELOTS learning module titled A Rainforest with a Gold Chain, the Impacts of Gold Mining on the Trophic Web of an Amazonian Rainforest of Southeastern Peru, based on this research. The module demonstrates how gold mining affects species throughout the food chain by a bioaccumulation of mercury in one of the most biodiverse forests on Earth. Ursula and Roxana have decades of experience working in this area, and Jacqueline and her colleagues developed ways to study the biogeochemical cycling of mercury and the impacts on local social structures and economics.
Hello, my name is Ursula Valdez. I'm a tropical avian biologist, originally from Peru. So my time in the field studying birds of prey, hawks, eagles, falcons, and mainly in the rainforest of the Peruvian Amazon. I'm very excited that I had a career that more than work feels like a lifestyle that I really love. I'm driven by curiosity about the natural world, and I tried to find patterns that could explain at least a little about how organisms live. I'm fascinated by the life of birds in particular, not only by the different colors they have, but also what they do. How do they find food? Where do they go? Where do they nest? How do they interact among their own species and with other individuals of other species? I'm so lucky that I have the opportunity to continue research in the tropical rainforest of the southeast Peruvian Amazon, where I go constantly. A region known for its great biodiversity, where we have more than 800 species of birds, and also many species of mammals, amphibians, reptiles, not to mention records for insects and plants and many more. Learning about the rainforest, the ecological processes, the organisms that live in there, the connections and mechanisms that help these ecosystems function. It's very important, not only to maintain biodiversity and sustain it, but for the entire world. Very fortunate that I was exposed to the natural world since I was very young, and I was encouraged to explore it. My parents were very supportive of my curiosity and provided great education. And I had great teachers and mentors who inspired me and gave me exciting opportunities to learn in the classroom and mainly during field classes and fieldwork training. There have been challenges also, such as a limitation on the field opportunities availability that there were in the past, or at least not everybody knew about them. We needed to be constantly in search of those, but in many ways, No internet available at that time that was that easy. Field biology and going to remote places was highly dominated by men also. And it was a little hard to break those preconceptions that women were not for hard work in the field, which is the career that I wanted to have. I also spent doing research with birds of prey, a field dominated by men, even more in this case. My research has been primarily focused on the ecology and natural history of forest raptors in the Peruvian Amazon and Madre de Dios. I address questions associated with their use of habitat, breeding, and foraging ecology, and the impacts of humans on this species. Much recently, I have extended my efforts to monitor bird species abundance and distribution and the structure of full bird communities and how they're impacted by habitat, loss, and deforestation, and other human activities. In Madre de Dios, one of the main reasons for habitat loss is illegal gold mining that has detrimental impacts on biodiversity, ecosystems, and humans. This is one of the remains of illegal gold mining site. As you can see, in the past, this used to be a rainforest, just like you can look over there in the farther back, it was all rainforest. We're very close to the Madre de Dios River, and one of the things that happens is that people will come and make this destruction of soil and sediment to remove gold and powder from the sediments, creating this big pool. And then later, after they finish extracting all the gold, which comes in powder, then they abandon them. So right now, these are places that have been recolonized by grasses on the upper part, and then lots of other vegetation remaining, all this water that birds are using actually and creating this habitat. But at the same time, all this water is polluted with mercury. How much that mercury accumulation is affecting species? That is a question that other colleagues are trying to answer. My work is not only about studying birds and teaching about them, but also learning from collaborators such as my colleague Jacqueline Gerson, the main author of the research we discuss in our gold mining module.
Hi, my name is Jackie Gerson, and I'm an assistant professor at Cornell University. And I wanted to share a little bit about the work that I have done in the Peruvian Amazon in the past. The motivation for my work was thinking about trying to understand where mercury from artisanal and small-scale gold mining was ending up. I know that artisanal and small-scale gold mining is responsible for about 1/5 of all of the gold produced worldwide, so a really large proportion of the gold. If you're wearing any jewelry like I am, it likely came from artisanal and small-scale gold mining. And the same process is responsible for about 1/3 of all the mercury emissions that occur globally, more so than any other source, including coal combustion. And that's because miners take liquid elemental mercury and they mix it with soil or sediment that they've mined in order to extract the gold. The mercury forms a coating around the gold, which makes it really easy for them to then burn off the mercury, leaving behind solid gold that they can sell in the world market. But as I said, this is a huge emitter of mercury pollution globally. And we know that this mercury is ending up in water and having an impact on fish and human populations that are eating this fish. But what I was really interested in understanding was whether there could also be impacts on the terrestrial ecosystem. And so the work that I set out to do was to determine, is this mercury also ending up in terrestrial soils? And if so, could terrestrial organisms also be exposed to high mercury loads? And interestingly, what I found was that in a really remote forest in the Peruvian Amazon, far from any industrial activity and biodiversity conservation area, we're seeing the highest ever reported concentrations of mercury in some of the songbirds there. And I know from talking to researchers who are still conducting work in this area that we're continuing to see really high concentrations of mercury in pretty much every organism that's being examined. And so this is really interesting, important scientific finding, and really shows us the importance of trying to protect our ecosystems and reduce mercury emissions from artisanal and small-scale gold mining.
Doing research is a fascinating activity, but it's never easy or a straight line to an answer. It's taking Jackie, colleagues, and students to develop even more questions from the results they already have. So many things are not answered at once. You need to keep trying. I myself have spent several field seasons trying to figure out the movements and distribution patterns of some of the species of birds that are the most common. And it still is difficult to find out where exactly they go at certain times of the year. With the data about the birds in Jackie's research, there is so much to learn and understand. How is that birds in remote areas, those that we know have smaller ranges and they are not even near water, how is that they get mercury in their tissues? So much to learn and many opportunities for students like you to discover in the future. Come learn with us. You can always contact us and be part of the effort to learn more about this amazing ecosystem here in Madre de Dios of Peru. Tropical avian butterfly.
This is great. So now we will go to Champaign-Urbana, Illinois, where Professor Krista Monroe, now at the University of Illinois, used Aaron and Carlos's module in the classroom at her former institution, the Landmark College, with her undergraduate chemistry students, and then authored an implementation plan for instructors who'd like to use the module in the future, with an emphasis on accessibility for neurodivergent students. Her student, Anastasio Bonhomme, also helped to adapt this module. This video is a little bit different. It focuses on the educational experience of students. These OCELOT modules are highly versatile. They could be implemented in a single day or expanded into multiple weeks of a semester, giving students chances to think critically about the content and to generate really impressive discussions and products.
Hi, my name is Krista Monroe. I am currently in transition from a role at Landmark College, which is in Southern Vermont, teaching chemistry, to a new role at the University of Illinois Urbana-Champaign as an educational research associate for their Center for Innovation and Teaching and Learning. I've spent the last 5.5 years teaching at Landmark, which is one of 2 institutions in the United States that exclusively serve neurodivergent students with learning differences such as autism, ADHD, and dyslexia. And I hold a PhD in chemistry. And my career has really focused on inclusive STEM education. I was really drawn to teaching at Landmark because I am very passionate about science outreach and teaching in non-traditional ways. And I've learned so much, not only from my colleagues and mentors at Landmark, but also from the students and the student voices, helping them to discover their strengths and build confidence. I would say that an important experience or person that was a turning point for me was my experience while I was getting my PhD at Princeton University with my mentor, Dr. Dan Steinberg. I was able to do a science communication fellowship with him, which allowed me to see what happens beyond the lab. I'm in a lab now. I've been working, teaching chemistry and being in the lab, but it was really helpful for me to see what other careers I could pursue. And I'm still in that process of exploration.
My name is Anastasio Bonhomme. I am about a semester away from getting my bachelor's degree in biology. And I've been interning with Dr. Monroe for about 7 months now. Starting in summer of 2025, we were working to help develop author guidelines for making online teaching modules more neuro-inclusive. That is to say, inclusive to every neurotype, not just divergent or typical, but everybody in the whole spectrum. I think one of the most important aspects that led to this career path would be the access to mentors and, role models who are able to direct you to these kind of opportunities. linking with the right professors at my school definitely had a big impact on my trajectory through the system, and I probably wouldn't be here without them.
The biting into science cookies module from the OCELOTS group into the Principles of Chemistry 2 class. There was a twofold module engagement. We first started by having the students work through the online modules, and then we coupled that with a hands-on experiment exposing the different types of seeds to some chemical contaminants such as wine tannins and hydrogen peroxide, looking at the effect on germination rates and sprout length. So the OCELOTS module introduced ecological and chemical principles behind seed dispersal. The hands-on germination experiment wasn't quite related, but it allowed students to make some connections between the environments that seeds are exposed to and the impact on germination and growth. The experiment was pretty simple. These are the different materials that you would need, and you can substitute any types of chemicals that you have in the lab. You just need a Ziploc bag, a sandwich bag, and some paper towels along with the seeds. We used the Wisconsin fast seeds, which actually ended up growing a little bit too fast. We had results within 24 hours. So you could slow that down and use regular seeds and probably get about a week to germinate. And what's also nice about this is you don't need a special environment like extra lights or anything like that. For the core themes of the module itself, from OCELOTS, covered mutualism, chemical signaling, and seed dispersal and human impacts on germination. The goal of implementing this in the chemistry class was to bridge chemical theory with real-world ecological applications and ultimately introduce students to thinking about sustainability. Many of the modules that are done with OCELOTS and are implemented are done outside and in biology classrooms. What makes this module unique is that we implemented it inside in a chemistry lab. There are a lot of differences in terms of thought process that ecologists have compared to chemists, and I think that exposing students to the different ways of thinking is really important and different ways of problem solving, especially because it allows them to kind of say, oh, you know, maybe being inside in a lab isn't the thing that I want to do, or maybe it is. And so it gives them that opportunity to do that exploration. I think that the overall feedback I received from students is that they were really excited to see the real world application and truly see it because they were able to experience it through the multimodal aspects of the videos and different things like that, and hear directly from the scientists.
I think one of the most thought-provoking elements was in general how well illustrated some of the concepts were that one may not have considered. Reviewing the modules did, I think, help illustrate how much of an iterative process research is, considering these were materials that had already been created, and it was our job to go in and review and find flaws, things they could do better, things they already did well, to try and pivot or capitalize on strengths that were already there.
That's great. I love to see the creativity and application of these modules. For our last stop, we'll go on a trip to the La Selva Biological Station in Costa Rica, where Erin Kuperwitz will speak about research covered in the OCELOTS module titled Biting into Science Cookies. Are the seed fates determined by seed size or chemistry? This module is authored by Erin and Carlos Garcia Robledo, and their module highlights another important aspect of chemistry and ecology. Through this, you'll get to know an intriguing rainforest mammal, the Central American agouti, while integrating aspects of seed ecology, animal behavior, and chemistry within an experimental setting in a Costa Rican rainforest.
Hi, everyone. I'm Dr. Erin Kuperwitz, That's me in a tropical wet forest in Costa Rica. Unfortunately, the soundtrack for this clip got corrupted, so I'll continue my introduction as a voiceover. I'm a curator in the Connecticut State Museum of Natural History, and I also manage the vertebrate collections in the Department of Ecology and Evolutionary Biology at the University of Connecticut in the USA. I call myself an ecologist and a mammologist, and I conduct research mostly in the tropics, in the neotropics or the Americas, and also in museum collections using specimens that have been collected from the field. I love getting to work directly with different species of mammals and plants. I grew up outside of Los Angeles in California and spent a lot of time during my formative years in museums and zoos, but not really in the wilderness. My family wasn't really into camping or hiking. However, I was able to take a tropical ecology field course when I was an undergrad at UC Santa Barbara, and the rest was history. I was completely hooked on tropical biology. Aside from doing research, for my job, I interact with other scientists, with students, and with the broader public to promote the use of collections in outreach, education, and research. Hi again, everybody. I've changed my scenery and I've come here to the University of Connecticut's Botanical Conservatory. So this is our living collection of plants, and I'm here in the tropical rainforest area, not in the field. I'm actually in Connecticut, so not in the tropics, but it mimics the tropics. This is also a good point to say, you guys, wherever you are, if you can't get to the tropics easily, seek out a botanical conservatory or some kind of botanical garden, or maybe you even have a living collection of trees or plants on your campus if you're at a university, and check it out and visit. For the module, Biting into Science Cookies, our overarching question was, are seed fates, so seed survival over time, affected more by seed size or by seed chemistry? And this is in the context of biotic interactions. So interacting with biological entities, in this case, animals that are moving and eating and dispersing seeds. This is a really big question in the field of plant-animal interactions ecology. So what traits of plants, in this case, what seed traits are affecting seed survival, seed escape from predation by, in this case, mammals, but it's also analogous to things, other animals that are eating seeds like insects. We have a lot of beetles and even ants that consume seeds as well. So this is a huge question in the field of interactions biology, seed survival and why seeds survive in really complex environments like those really typical of tropical lowland forests. What kinds of data did we collect and how did we collect those data? So we're Muddy Boots ecologists. We're on the ground manipulating seeds. We created these science cookies, which I also call artificial seeds, out of a non-toxic polymer clay and peanuts that we bought from the store. We created a fruit that mimics drupaceous fruit, a drupe, and kind of put peanuts inside a clay football, basically. We attached a string to these seeds, like a 50 centimeter length of fluorescent pink twine, and we used camera traps to record what species, or what individuals even, were interacting with which seed. We changed the sizes of the seed, so we had 3 different size classes. We were able to replicate creating hundreds of these little cookies for agoutis to interact with, and we looked at how far agoutis were moving things in space. We would go back every 2 days or so to record which species, or which individual seed was moved where in space. And was it eaten? Was it a predation event? Was it buried under the soil by an agouti in a dispersal event, so a scatter hoarding event? These behaviors have really serious implications for seed survival over time. The main thing we found was that decision-making by agoutis is very complex. They weigh a lot of different plant traits when they're deciding what to do with a seed, to eat it immediately, move it really far away, bury it in the soil to protect it from other seed predators. But in all of that, they did not care about chemistry whatsoever. Size of seed was the only thing that mattered when they changed their behaviors. So small seeds tended to be eaten quickly. Larger seeds were moved really far and buried in the soil for future consumption. Concentration of tannic acid in every treatment of seeds did not matter at all to agoutis. They did not care about the chemistry. Either they didn't really consume the part of the seed that had the tannic acid, or it was just a bitter flavor and didn't bother them enough to care. Hi again, everybody. I'm coming to you now from a different location. I'm in a different type of collection here on the University of Connecticut campus. These are the biodiversity research collections in the Department of Ecology and Evolutionary Biology at the University of Connecticut. So this is where we keep our research specimens. We have taxidermy, like this giant flying squirrel behind me. We have osteological collection, like these bones that you see here. And we also have round skins. That's the majority of the vertebrates that I take care of here in the research collections as the collections manager. This collection is here and accessible to anyone who wants to conduct research using our specimens that are collected from all over the world, including a lot of specimens from the tropics and from Costa Rica. So this is a great place to seek out if you want to do any kind of research near your own campus or on your own campus. Check out if your campus, if your school has a collection, has collections of animals, of plants, like an herbarium. A lot of places have entomology collections, so pinned insects for the most part. But here at UConn, we have a great collection of vertebrates. So how did I come up with this research question to think about how seed size and seed chemistry affect decision-making by potential mammal seed predators and seed dispersers? It came from looking at things in the field, in situ, we call it, in the place where it exists. So walking around the rainforest of Las Cruces Biological Station, I saw a ton of agoutis eating a lot of different types of seeds. Again, tropical rainforest, pre-montane rainforest. There are so many species of large seeded plants, seeds with different sizes, seeds with different chemistries and shapes that agoutis tended to eat. And I wanted to know what affected their decision making and how did their decisions affect the plants. So kind of this circular idea of what are the animals doing to the plants, what are the plants doing to the mammals, and how is that affecting both of those types of communities. Research is very rarely linear. You'll start usually with one hypothesis, one group of questions, and then as you're getting more evidence, you find new questions to ask, or something's not working. Some piece of equipment broke in the field. The animals aren't there when you need them to be there. The seeds, the plants aren't there in enough numbers for you to ask the questions you want. You have to be really creative as a scientist, and you have to follow what works. That's what I always tell my students or the other researchers I work with. We'll start out one way, but have the adaptability to ask other questions and to follow other lines of research. When one line isn't working, know when to leave it. All right, that does it for all that I have to say today. Thank you so much for paying attention. I hope that you come away with a greater appreciation for large, charismatic, tropical potato-shaped mammals and what they do with plants. If anything that I talked about here today was interesting to you, send me an email, and I hope to see you in the jungle sometime. See you later.
I love it. Well, before we move on to meeting our presenters for the round of Q&A, we do have a favor to ask, actually two. First favor is please write your questions into the Q&A. That will really help us. And then please take a screenshot or photo of this slide with survey information. It's really important for us to gain insights about your experience in this webinar, both for our reporting, but earnestly to help us improve these events. So instructors, please watch for emails about the consent form and post-event survey from external evaluator Liesl Seriak at Measurement Resources Company. The consent form needs to be completed before doing the survey. Students, we'd love to get your feedback from a 2-minute survey at the URL or the QR code that you can see in the slide. So, and the URL is in the chat. Please wait, though, to take this survey until after we do the Q&A portion of the event, and then we hope you'll just do it as soon as you can. Taking a photo now of the QR code or copying and pasting that URL from the chat into an email or into a document on your computer is recommended. All right, so now it's time for your questions and for the panelists. So please, I see we've got a few questions going, but please post more questions using the Q&A button rather than the chat. Our college classes have a time constraint of 50 minutes. So we're going to prioritize those questions, those where they're not the non-anonymous questions. After that, we'll answer all questions from any attendee for the last 10 minutes. And the panelists are going to try to keep their answers short to about a minute so we can get to as many questions as possible. All right.
All right, welcoming our panelists on the screen in the flesh. Great to see everybody today. All right, to get everybody started, I'm just going to ask all of you the same question. And if you could just keep it kind of short, but we'd love to hear from all of you. So first off, let us start in the order we saw you all. So we'll start with Ursula, then Kristen, and then we'll move on to Erin. So can you tell us or remind us what drew you to field research, tropical biology, or chemistry in the first place?
All right, so thank you, Kerry, and thanks everybody for being here. I think I was a very lucky child. I got the opportunity to go many times in expeditions with my parents in Peru. And then I have awesome mentors, including one that is here, Martha. I have had the opportunity to do a lot of field work thanks to opportunities in my college. And then later I took every single field job that I could. So just the connection in being in the field was a very important aspect to guide me into discovery and curiosity and things that then they turn into a science career.
I will reiterate Ursula's comments about mentorship. It's so important. I failed my first chemistry class in high school, and without mentors, I would not have persisted in chemistry, and I definitely had a lot of bumps along the way. But I think it was actually the math that drew me to chemistry and the problem solving that is involved in it. And even though I'm not currently a chemist, I see that problem solving training utilized every day in my work.
Awesome. So mentorship is huge, but honestly, I fell in love with the tropics, especially when I was a little kid reading like National Geographic magazines. Zoo books were a big part of my life. I played like computer games that were educational, like Amazon Trail was a big one. Those kinds of things I got to, again, like I mentioned in my video, museums and zoos and aquaria. I lived in like the suburbs outside of LA. I didn't have a lot of access to wild spaces. And so kind of once I was in college and old enough to get out to the field, I went every chance I got. And I had a lot of mentors, instructors, people that I worked for that really encouraged that and kind of pushed me along to get to the field in any way possible.
That's all fabulous. All right, first question from the audience is, I think actually might be interesting to hear both from Erin and Kristen for the field experiments and the lab experiments. The questions from Adria Gonzalez Carlson. What type of acid was used in the artificial seeds?
Sure. It was tannic acid. So I was trying to find something that wasn't especially toxic to animals. I didn't want to hurt them. But it's something that gives like a bitter flavor to things. So you have tannins in like wine and coffees and like bitter stuff. So it was more like an aversive kind of thing. So tannic acid of basically the highest concentration I could get within the seeds or the fruits was like 15%, I think.
We on that same vein, we were using the Wisconsin fast plants, but we were exposing them to both hydrogen peroxide and tannic acid to see the impacts of germination. But you can really test it with anything, and it's kind of fun to have the students come up with different hypotheses and design the experiments with that.
That's great. I think we have some chemists in the audience. Okay, great. So let's move on to our next question. So this one is going to be for Erin. I noticed that you made some drawings for your modules. How have you managed to include being an artist into your biology career?
Ah, lovely. I also have my little pin that I made, my little goodie. So I use art as kind of like a creative outlet, but I also publish I try to like sneak drawings in when I publish things as well. It's great for outreach. It's great for presentations. But sometimes publications, the editors are pretty cool with it usually. So again, it's kind of like a creative outlet, but it gives me a way to think about things and kind of visualize different components of an experiment. Like I need that like hand brain link a lot of times to really make sure I have all my ducks in a row and like get out to the field and work on things. So it's kind of like to show people things, but also to help myself think about different parts of science.
That's so awesome. So I've got a question for Ursula Valdez. What prepared you the most for collaborating on research projects like the one you presented about today?
Mhm. Well, I guess the interest of expanding our own, we know certain things. We don't know everything. We cannot solve every question. We cannot just do everything ourselves. And the fact that we have so many people working in the same systems or even in other parts of the world that are addressing in a different way, probably the same questions, that brings an incredible value to understand a pattern that you've been curious about. And that it could be, a much wider perspective of the situation that you're trying to understand. So that it's a very important aspect of being a good scientist because we have to recognize our own limitations and how we can do better when we can join other people in understanding and solving. I think it was Kristen who talked about the problem solving skills. It works better when you have more people and more brains to tackle a question.
Lovely. Kristen, this one's for you. Okay, so this person wants to know if you have any advice for other educators who want to make resources more accessible for neurodiverse students, and also if you have a favorite resource.
Okay, so I think that this is such a broad question, but um, my favorite resource is actually my students, asking my students what works best for them, which I know you can't always do because there are a lot of invisible disabilities out there. Another thing, I'm just putting this in the chat, is Brown's model of executive function. So executive functions are the tasks that we need to go through to complete assignments. I have historically had a lot of students that don't turn in work, and trying to figure out what is the barrier to them not turning in work. And Brown's model, even though it was more developed specifically for ADHD, it is widely applicable, in my opinion, to a lot of different types of students, neurodivergent and other. And so thinking about, as you're designing your assignments, how you can minimize the barriers that students have to go through to complete the assignment is probably my go-to if I had to like boil it down to one thing.
That's fabulous. I'm gonna use this. So I am going to pull the next student question, and just mention, I know some students may have to leave, and if you do, just thank you so much for joining us. And we will be emailing with follow-up information, and but I'll continue on for now with the next question. So the question is, mentorship was mentioned. As a student going to look forward to doing more research in the field, where should a student look for a mentor? So I think really that's to all of you. So maybe very briefly, you could each address that.
Sure, I'll go. I guess look for all the, when you take a class and you like a class and you meet the professor, ask the professor. That's one of the things that, many of them are doing research themselves, and others, they can connect you with someone else. Look for opportunities in your department. Many departments have the undergrad services that they have people who actually have plenty of resources for you. And ask people who have been in the field. I think that would be my advice. Ask those students, take a study abroad programs to start with. That's an incredible opportunity that you may not know that you want to do research, but take a taste, right? Go somewhere that is different to what you were thinking before, something that can expand your horizons, and you never know when it's going to be that time. Click in your trap.
All right. So I have two questions really quick. So we have a professor in the audience who said that they've never heard of Brown's hierarchy of executive functions and would love it if you could give an example of how to create assignments that remove or reduce barriers for completion. So if you can answer that, or if you have any resources to put in the chat, Kristen.
So just very briefly, one of the classic things is activation, right? We're all scientists, so I can say my students have a high activation energy. And so if there are things that you could do, like getting students started at the end of class on an assignment and helping them brainstorm, even using AI as a thought partner to help them brainstorm, especially completing the tasks that aren't necessarily assessed directly. Another one is there's a lot of anxiety in completing assignments. And so if you can, so this is more of just like broad course-based thing, course policy things, but if you can give students the opportunity to, if they turn it in early, then they have the opportunity to revise it, giving them a little bit of flexibility there, because that anxiety of not knowing whether they're doing the right thing can really cripple them. And so having that opportunity to get feedback and see what is actually happening in their heads can be really valuable. I'm also happy to chat more about it, so I can just put my email in the chat if anybody wants to follow up with me. But great question.
I love that we have a discussion going already. That's fabulous. Okay, great. I am going to ask a question that's unusual. This is a question for Martha. We never get one for our moderator. So this person wants to ask Martha a couple questions. First is, what fostered your interest in ocelots specifically, and what do you think is so special about the animal? I think they might be thinking of ocelots modules and ocelots the animal. So ocelots as a title actually stands for a longer phrase. But they also mentioned that there's an incredible documentary called Wild Cat, and that's one of the main things that got their interest in working in animal conservation and rewilding programs started. So just wanted to field this to Martha in case you had anything to say on that.
Yeah, I think that's just fabulous. I'll just say my own interest in ecology and the tropical ecology was started through documentaries and nature programs that I watched as a kid, suburban kid. So I really feel you there. I haven't seen that one, so great tip. And I am also interested in ocelots because ocelots are beautiful. I walked by one once in the early morning when I was going to start my work on birds, and I could just feel it there. And I turned around and there it was, looking at me, waiting for me to pass. So that is a really exciting feature of being in the field. And I find that here in Washington State too. You just see such interesting creatures, so many different behaviors. It's really fun. So get out there. It's great. Okay, well, I'm going to ask a question of Ursula, if that's all right. My question is actually partly from my experience with my students. You've shown in the module some really big impacts from gold mining. And so I know a lot of people are interested in understanding, like, is there anything I can do here in the US that might make a difference?
Yeah, I mean, we all have a role on this gold mining issue. So even if you're interested in the ecosystem, if you're interested in people, if you're interested in both, which I hope, the thing is that not only is destroying habitats and ecosystems is also affecting a lot of people. So the gold market, it's a huge market, and as you hear in the video, 1/5 of that comes from illegal gold mining, artisanal gold mining, that in many places is illegal, in particular in this location in Peru. 90% of the operations are illegal, and those are affecting a lot of people. So research, first of all, where, the kind of things that you're buying, where they come from. With gold, it's very difficult actually to track. It's not like, I don't know if you remember, there was something about diamonds that you can actually track diamonds, where they're coming from now. Unfortunately, so far, we can't, but hopefully some researchers will find a way. But because the gold is melted and then it can be joined with anything else, you lost the tracking. So it's impossible to track where is it coming from. But still, you can maybe reduce the gold use in some of the unessential things. And that's one of the things. The other part is support activities that actually are alternatives for people in those places that aren't exploiting or doing gold, because they don't have any other option. So now there's ecotourism companies. Research well if you're going to go there. Figure out there are more companies and conservation organizations offering alternative products that come from the forest, that are extractive opportunities for people, but they're not having the detrimental impact that gold mining does. And support research, support efforts and conservationists that are right now working to recover those areas and working with people. So being informed, it's a first step about the kind of things that you can do. Learn about this module, learn from your professors, talk about that. And yeah, I will leave it in there. Thank you for asking, Martha.
Thank you so much. Well, our hour has flown. It has been so great to meet the three of you, to watch the videos that you put together and learn so much. So thanks. Let's everyone thank our panelists, Ursula Valdez, Kristen Monroe, and Erin Cooperwitz. And also our video presenter and researcher, Jacqueline Gerson and Anastasio Bonhomme. We also want to thank the Organization of Biological Field Stations, the OCELOTS Network, and OCELOTS 2, and the Center for Environmental Inquiry at Sonoma State University. You can learn more about all of these amazing things that the organizations are doing at the links that Kerry is pasting into the chat right now. And lastly, thanks to the National Science Foundation for supporting several aspects of this work, and to a lot of wonderful people behind the scenes who make this all possible. We are going to be emailing all of you who participated today in the next few days with follow-up answers to any questions we were unable to address, a link to the recording of this event, a link to the survey, which, remember, it's a very short one for students, and many of the faculty members here will have already received a consent form with a longer survey that we really hope you'll take the time to fill out. I filled out a whole bunch of these. They're really actually pretty fun and thought-provoking. So thank you in advance for this important way of tracking and assessing the success of these events, as well as helping us make them better in the future. We hope to see you at some future Live from the Field events that are currently being planned for the 26-27 school year and beyond. Please share these as well as the recordings of past events with fellow instructors, students, friends, and colleagues. And thank you and goodbye from the field. And panelists, let's wave. Okay. Bye.